Hi everyone, today we're going to be solving AQA, GCSE Biology, Higher Tier, Paper 1H. In this particular video, we're solving June 2018. We'll be solving the entire question paper, and guys, all the answers and all of the options of the answers will be provided. So, just watch them thoroughly, alright, from the beginning till the end. Eating food containing Salmonella bacteria can cause illnesses. Two symptoms of infection by Salmonella are vomiting and diarrhea. What causes these symptoms? So here we have to apply the knowledge that bacteria produces toxins or poisons. All right, so we will use that particular knowledge and our answer will be, so what causes the symptoms? Our answer will be that toxins are secreted by the bacteria. Give two ways a person with a mild infection of Salmonella can help prevent the spread of the bacteria to other people. So when a person has a mild infection of Salmonella, preventing the spread of the bacteria to other people will involve such techniques where we will prevent the bacteria from going from one person to another person. So for example, when the person will use the washroom, after using the washroom, the person should wash the hands after using the toilet when they are sick. The person must not prepare any food while they are infected for himself or for others. It is better that the person isolate himself. The person should disinfect their clothes because with their unwashed hands or somehow they can also contaminate their clothes, right? So that can be one of the ways. And the person must not share their utensils and cutlery with another person. In very serious infection of Salmonella, a doctor can prescribe drugs to kill the bacteria. What type of drug can the doctor prescribe to kill the bacteria? Since we are trying to kill a bacteria, so antibiotics will be the right type of drug to kill it. A person with AIDS may take longer than a healthy person to recover from a Salmonella infection. Explain why. When a particular person has AIDS, then the person's immune system is damaged or weakened. Since the person's immune system is weakened, white blood cells cannot kill the bacteria or, in this case, the Salmonella as effectively. Salmonella bacteria can be transmitted from chickens to humans. Chickens can be vaccinated to prevent the transmission of Salmonella bacteria to humans. Suggest one other way farmers could prevent the transmission of Salmonella from chickens to humans. So the farmer can have many different ways to prevent. One of the method will be the farmer can give the chicken antibiotics so that the chicken by itself is free of Salmonella bacteria when it is consumed. The other one that the farmer can do is that the farmer shouldn't sell the infected chicken or the infected chicken eggs to the public so that people don't get sick by eating those infected products. The farmer can also keep the infected chickens isolated indoors so that it doesn't get the chance to mix with other chickens and spread the Salmonella to other chickens. The farmer can also slaughter the infected chickens, all right, and chlorinate those wash them with chlorinated water to get rid of the Salmonella bacteria in them and thoroughly cook them before consuming them. A restaurant owner employed a scientist to test the effectiveness of two kitchen cleaning liquids. The scientist took samples from the two work surfaces before the surfaces had been cleaned with the cleaning liquid and after the surfaces had been cleaned with the cleaning liquids. The samples were then analyzed for a number of bacteria they contained. So we can see in the sink with the cleaning liquid A, before cleaning, the number of bacteria is very high. All right, it is almost 140, and after cleaning it goes down only to 25. So we can say that after cleaning liquid A also has the same effect with the table, goes pretty low after cleaning, and cleaning liquid B, we can see it decreases even more. It's more effective than cleaning liquid A, and in terms of cleaning the table, we can see the number goes very low in comparison with cleaning liquid A. The question asks, which cleaning liquid is more effective? Give a reason for your answer. So definitely cleaning liquid B will be more effective. Now, the reason that we will give the answer is that there is a greater reduction in the number of bacteria after cleaning in both locations with cleaning liquid B. The scientists investigated the effect of cleaning liquid A and cleaning liquid B on the Salmonella bacteria grown in a laboratory. Figure 2 shows the way the investigation was set up. We can see Salmonella was grown on agar, and then we had filter paper that was soaked in water with cleaning liquid A and cleaning liquid B. After 48 hours, the scientists calculated the area around each paper disc where no bacteria were growing, and they were incubated for 25 degrees Celsius for 48 hours. With water, there was no clear zone, and with cleaning liquid A, there was 11, and cleaning liquid B, there were 13 cm square of clear zone. What measurement would the scientists need to take to calculate the area where no bacteria were growing? So the scientist needs to take the radius. I mean, if this is the clear zone, the scientist needs to calculate the radius. Once the scientist knows the radius, the scientist will be able to calculate the area. So the scientist needs to take the radius of the area with no bacteria growing. Give one change to the investigation that would allow the scientist to check if the results are repeatable. The scientist needs to repeat the experiment and look to see if the results are similar. The scientist showed the results to the restaurant owner. Both cleaning liquids cost the same per dm cube. Suggest one other factor that the restaurant owner should consider when choosing those cleaning liquid to use. Since both of them has the same cost per liter, then other things that the scientist should take into account is whether the cleaning liquid solvent that he is using has any health effects, like side effects or not, and the restaurant owner can also test the effect on other types of bacteria and pathogens which they didn't use in this experiment, all right, to see their effectiveness on those, all right, bacteria and pathogens. And then the scientist, the restaurant owner can also ask the scientist, or maybe can also test the ease of the use of that particular cleaning liquid. The restaurant owner can also see the dilution factor of each of the cleaner versus the cost and the time the cleaner is effective for. So all of these are possible answers. However, for 1 mark, we only need to write one of the points. Metabolism is the sum of all the chemical reactions in the cells of the body. One metabolic reaction is the formation of lipids. Give one other metabolic reaction in cells. So when a question like this is asked, you have to understand that any chemical reaction taking place in the body can be regarded as a metabolic reaction. So for example, respiration, formation of proteins, formation or breakdown of glycogen, breakdown of excess protein to form urea, photosynthesis, formation of glucose in plants, all of these are regarded as metabolic reactions. So I will just give you a few examples, and the ones that I have given you as in my answer, all right, I'll try to mention them in points here. So mean metabolic rate in kilojoule per meter square per hour for males and for females is given from age 5 to 45. We can see in males, all right, the mean metabolic rate in kilojoules per meter square per hour, it's the highest when we were younger and decreases as we become older, males. And for females, what do we see? For females, we can see that it is the same when they are also younger. However, it decreases much more rapidly. As you can see, by the age 25, it's 39 for male, whereas it's 35 for a female. And then it doesn't decrease any further and remains 35. So the question says, what two conclusions can be made from the data in table 2? Let's see the conclusion answers. As age increases, mean metabolic rate of males and female increases. We can see in both cases, it decreased from 53 to a lower number. So this is a wrong statement. Males have a higher metabolic rate than females after 5 years of age. It is true, because at 5 years of age, they both have the same metabolic rate, but after that, throughout the life, males have higher metabolic rate than female. So this is correct. Then the next point, the mean metabolic rate of females decreases faster than males up to 25 years of age. That's true, because we can see at 25 years of age, females have a lower metabolic rate and much lower than that of a male. So we can say that it decreases faster. We can agree on that particular point. The mean metabolic rate of males and females decreases more quickly after the age of 35. Now we can see for female after the age of 35, it doesn't decrease any further. There is no decrease. So this is a wrong statement. There is no relationship between age and mean metabolic rate. This is also wrong because there is a relationship, and that is a negative relationship. As the age, as the age increases, we can see that the mean metabolic rate decreases. Calculate the percentage decrease in the mean metabolic rate of males between 5 years and 45 years of age. So in terms of percentage decrease in metabolic rate divided by original metabolic rate times 100. Give your answer to 3 significant figures. Okay, great. So we can see from the age of 5 years to 45 years. All right. So for males at the age of 5, the value is 53, and the age of 45, it's 36. So since we have to do percentage decrease, so we will do 53 minus 36 divided by the starting point, which is 53 times 100. So it becomes 17 divided by 53 times 100, and thereby gives us 32.1%. Regular exercise can increase metabolic rate. 2 people did 5 minutes of gentle exercise from rest. Table 3 shows the effect of the exercise on their heart rates. We can see time in minutes and then heart rate in beats per minute. So from 0 at rest to 5 minutes, we can see that the heart rate for person R increases up to 4th minute and then remains constant, which is at 99 it remains constant. But for the person S, the heart rate keeps on increasing and increases up until 5th minute, and you can see it goes to a much higher level, which is 132. Describe the 2 differences in the response of person R and person S to the exercise. So for the person R, we can say that the heart rate increases, and for person S, the heart rate increases too. But person R heart rate increases more slowly and compared to person S. Person R, the heart rate levels off at a certain pace, but for person S, the heart rate keeps on increasing until 5th minute. For person R, the heart rate rose overall after 5 minutes of exercise. Alright, so for person R, the heart rate rose less overall after 5 minutes of exercise, whereas for person S, it increased way too much. So the answer can be written as follows. Complete the line graph in figure 3 for the person S. You should add the scale to the x-axis and label the x-axis. So x-axis always is the independent variable. Always remember, x-axis is the independent variable. So what is the independent variable here? The independent variable is for us for the time in minutes that we chose to find out their bits per minute. 0 is already labeled. 1, 2. So total number of time is 5 minutes. We labeled it, and we're going to write time in minutes in the x-axis. Time in minutes. So for 0, 78, 100, 110, we will do the labeling. After 5 minutes of exercise, the heart rate of person S was 132 beats per minute. When person S rested, the heart rate decreased steadily at a rate of 12 beats every minute. Calculate how much time it would take the heart for the person S to return to its resting rate. So the person S had a maximum heartbeat of 132 BPM, well, starting from 78. So what we need to do is we need to do 132 minus 78, and we need to divide it by 12. Now this will give us an approximate time of 4.5 minutes. So 4.5 or 4.5 minutes. A student made the following hypothesis about the heart rate of smokers and non-smokers during exercise. During exercise, the heart rate of smokers increases more than that of the heart rate of non-smokers. Design an investigation that would allow you to test this hypothesis. When designing an experiment of this type, alright, we need to take 2 groups of people. One of them will be non-smokers, the other will be smokers, and they must have at least 5 to 10 people for each of the groups. So make sure to have like, at least more is better, but let's say 10 people on each group, all right? And then we will get each person to do a particular exercise. For example, running in a treadmill at 5 kilometers per hour for 5 minutes. Now, we will keep few of the variables similar. What are the things we are trying to keep similar? First of all, we will keep the number of people in each group similar. We're going to use the same gender. We will use the person, those who have similar levels of activity already. We will use same age group, and we will make sure that they do not have any health issues regardless of them being smoker or non-smoker. And we will give them the same type of exercise and same time for the exercise. These are the controlled variable. Now, if you don't remember all of this as your controlled variable, don't worry, all right? To get a very high score or good score, we can at least mention 3 to 4 controlled variable. Then finally, what we're going to do is we're going to record the heart rate for each person before and after the exercise and calculate the increase in heart rate for each person after the exercise. Then we can calculate and compare the result for each group. So the answer should be written as follows. The circulatory system is composed of the blood vessels, and the heart. Urea is transported in the blood plasma. Name 2 other substances transported in blood plasma. Blood plasma carries both nutrients and waste products. So, for example, carbon dioxide, water, glucose, amino acid, lactic acid, all of this are carried in blood plasma. Some athletes train at high altitude. Training at high altitude increases the number of red blood cells per cm cube of blood. Explain why having more red blood cells per cm cube of blood is an advantage to an athlete. Having more red blood cells per cm cube means those red blood cells are going to have more hemoglobin. Thereby, the red blood cells will have more ability to carry oxygen and transport oxygen for aerobic respiration to the muscle cells. So more energy will be released by the muscle cells during aerobic respiration, and the athlete will be able to work harder. Which 2 blood vessels carry deoxygenated blood? So we know that aorta carries oxygenated blood, coronary artery carries oxygenated blood. However, pulmonary artery carries, deoxygenated blood, and also, vena cava carries deoxygenated blood. However, pulmonary vein brings oxygenated blood from the lungs to the heart. So this one carries oxygenated blood. Figure 4 shows 3 types of blood vessels. We can see blood vessel A. A is representing an artery because we can see it has smaller lumen and it has a thick muscular and elastic wall. B represents a vein because of the larger lumen and thinner, so this is vein, and a thinner muscles and elastic wall. Whereas C is only one cell, thickness, only one cell, and we can see that a lumen is only as about of a red blood cell, so definitely C is a capillary. The question says which type of blood vessel carries blood into the right atrium. Now, to carry the blood into the right atrium, we must bring it through a vein, so it will be B. B is a vein. Compare the structure of an artery with the structure of a vein. So guys, we have already done the comparison, so we can write the answer as follows. Arteries have a thicker layer of muscles and veins have a thinner layer of muscle tissue. Artery has a thicker layer of elastic tissue, whereas veins have a thinner layer of elastic tissue. Arteries have a narrower lumen, whereas veins have a wider lumen. Arteries do not have valves and veins have valves. Now, we have to compare the structure between artery and the structure of a vein, so we need to write in terms of comparison, and only 3 points are needed to get the marking point. However, I will give you 4 points. You can choose between them. Heart rate is controlled by a group of cells. This group of cells act as a pacemaker. Figure 5 shows section through the heart. Draw an X on figure 5 to show the position of the pacemaker. So the pacemaker is called the sinoatrial node and it is found on top of the right atrium. So here, anywhere here, if we place the cross, it will be correct. A patient may be fitted with an artificial pacemaker. What condition may be treated using an artificial pacemaker? So if the particular patient has an irregular heartbeat, too much or too less, then it can be treated with a artificial pacemaker. A student carried out an investigation using chicken eggs. This is the method used. Place 5 eggs in acid for 24 hours to dissolve the eggshell. Measure and record the mass of each egg. Place each egg into a separate beaker containing 200 cm³ of distilled water. After 20 minutes, remove the eggs from the beakers and dry them gently with a paper towel. Measure and record the mass of each egg. So we can see mass of egg without the shell in grams 73.5 and mass of egg after 20 minutes in grams 77. And then we can see this happening for egg 1, 2, 3, 4, 5. Okay, in every cases we can see the mass has increased. Another student suggested that the result for egg 4 was anomalous. Do you agree with the student? And we have to give a reason for our answer. So egg 4, we can see the mass has increased slightly to 73.1 starting from 71.6. So we will say yes, because the mass change of egg 4 is much lower than the others. Why? Because in most of the eggs we can see about an increase of 3.5 grams or 3 grams at least. In this case, it only increased by 1.5 grams or such. So we can say this is the anomalous result. So the answer will be yes, because the mass change for egg 4 is much lower than the others. Calculate the percentage change in mass of egg 3. To do the percentage change in mass for egg 3, 72.4, 75.7, we will write 75.7 and then we will subtract 72.4 and then we will divide it by 72.4, which is the starting mass, and then we will multiply it with 100 for the percentage. And once we do that, we get approximately 4.6%. Explain why the masses of the eggs increased. The mass increased because water enters those, eggs that didn't have the shell by osmosis from a dilute solution in the beaker to a more concentrated solution into the egg cell. From a dilute solution in the beaker to a more concentrated solution in the egg cell through a partially permeable membrane. Explain how the student could modify the investigation to determine the concentration of the solution inside each egg. The student can use 5 or more than 5 different concentration of salt solution in a beaker, and then the student can use these particular experimental value and plot the percentage change in mass of the egg in a graph. The student can determine the concentration where the curve crosses the zero, where the curve crosses the x-axis, and that will give us the concentration of the egg cell solution. Chicken eggshells contain calcium. Calcium ions are moved from the shell into the cytoplasm of the egg. Table 5 shows the information about the concentration of calcium ions. Eggshell, concentration of calcium ion in arbitrary 0.6. Egg cytoplasm 2.1. Explain how calcium ions are moved from the shell into the cytoplasm of the egg. So, first of all, there is a high concentration of calcium ions in the cytoplasm compared to that of the egg cell. So, the ions are moved from an area of low concentration to high concentration. That means it should be done by active transport, which requires energy from respiration. Plants can be infected by fungi, viruses, and insects. Aphids are small insects that carry pathogens. Figure 6 shows an aphid feeding from a plant stem. We can see the plant stem in the picture, and we can see the sharp mouthpiece that the aphid is penetrating the plant stem. An aphid feeds by inserting the sharp mouthpiece into the stem of the plant. Give the reason why the mouthpiece of an aphid contains a high concentration of dissolved sugar after feeding. The mouthpiece has pierced the phloem layer of the plant, and the phloem has high concentration of dissolved sugars in it. Plants infected with aphids may show symptoms of magnesium deficiency. Magnesium deficiency symptoms include yellow leaves, stunted growth. Explain how a deficiency of magnesium could cause these symptoms. First of all, the general understanding will be magnesium, since it is needed for the synthesis of chlorophyll. Without the lack of chlorophyll, the plant will have yellow leaves, and therefore less light will be absorbed by the chlorophyll, and as a result, lower rate of photosynthesis will take place. Therefore, the plant will produce less amount of sugar, and as a result, the plant will convert very less amount of sugar into protein for growth, and as a result, the plant will have a stunted growth. A farmer thinks a potato crop is infected with potato virus. Why? Potato virus, why? The farmer obtains a monoclonal antibody test kit for PVY. To make the monoclonal antibodies, a scientist first isolates the PVY protein from the virus. Describe how the scientist would use the protein to produce the PVY monoclonal antibodies. The process of making monoclonal antibody is similar for any protein that we need to make it for. So what is the process? First of all, we'll have to inject that protein into a mouse. Once we put it into a mouse, the mouse will combine that particular protein, will make antibodies against that particular protein, and then we're going to collect that particular lymphocyte that is producing the antibodies, and then we will combine it with a cancer cell to make a hybridoma cell. Once the hybridoma cell is made, then the hybridoma cells will make the specific monoclonal antibody, which is PVY. The scientist then can clone the hybridoma cell to produce many hybridoma cells that will make the antibody for the scientist. Cystic fibrosis is a genetic disorder caused by the cheat gene, where what molecules are genes made of? We know that genes are made of DNA, which stands for deoxyribonucleic acid. CF affects the cell membranes of the cells in the lungs and digestive system. What is the function of cell membrane? Generally, the function of a cell membrane is to control whatever enters and exits the cell. So we will say controls the movement of substances in and out of cell. In a person with CF, cells lining the lungs and digestive system create too much mucus. The mucus can block the duct leading from the pancreas to the small intestine, block the tubes leading to the alveoli in the lungs. Explain why children with CF grow more slowly than children without CF. Since the excess mucus can block the duct leading from the pancreas to the small intestine and block the tubes leading to the alveoli in the lungs. So first of all, for the blocking the duct leading to the pancreas brings the enzymes. So fewer digestive enzymes can enter the small intestine, and enzymes are needed to break down food. Therefore, less absorption of nutrients can occur. So less glucose then enters the bloodstream, and less glucose is then available for the respiration. Since there is a lesser absorption of the nutrients, so fewer amino acids can enter the bloodstream. So less protein is made, and protein, is needed for growth. Now, the blocking of the tubes leading to the alveoli in the lungs, we can answer, since less oxygen enters the bloodstream, less oxygen is available for aerobic respiration. So less energy is released and available for growth. I am including all of the marking points with different color codes, and you can choose any one of the 6 points. However, make sure that at least you choose 4 points for the digestive enzymes and 2 points for the oxygen availability. Table 6 shows information about people in the UK in 2015. People with cystic fibrosis median age in years 19 and the whole population median age in years 40. Describe how the median age of a group of people can be determined. We can put the ages in ascending or descending order, and then find the middle value, which will give us the median age. Suggest one way why the median age for people with CF is lower than the median age for the whole population. People with cystic fibrosis aren't able to live to an old age. People with a lung function below 30% may need a lung transplant. Table 7 gives information about people with CF in 2015. Lung function greater than 75, percentage of people with CF 22, 51 to 75, 72, 30 to 54, and less than 32. In 2015, the total number of people with CF in the UK was 10800, so 10,800. Calculate how many people with CF in the UK in 2015 would not need a lung transplant, since lung transplant is needed for any person, those who have a lung function below 30%. So we can see only 2% of the person with cystic fibrosis requires lung transplant. Since 98% doesn't need it, so we can say 10,800 multiplied by 98%, which will give us 10584 person will not need any lung transplant. Lung transplants from donor have risk. One risk is organ rejection. Scientists are researching how to solve the problem of organ rejection and hope to use stem cells to create healthy lungs. The healthy lungs can then be transplanted into cystic fibrosis patients without the risk of organ rejection. Describe how scientists may use stem cells to create healthy lungs that are not rejected by cystic fibrosis patient. So the process of producing a healthy lung using stem cells and producing any other tissues or organs using stem cells are similar. First of all, stem cells from the patient's bone marrow will be taken. Then the stem cells will undergo a process of genetic modification in it. All right, which we will refer to it in this way. We will genetically modify it so that we can fix the faulty cystic fibrosis gene. Then we will create a stem cell. Using the stem cell, we will create an embryo and then remove the stem cells from the embryo and stimulate the rest of the embryo to divide and differentiate into lung cells. Some people disagree with the use of stem cells because of the risk of cancer. Give one other reason why some people disagree with the use of stem cells to create new organs for transplants. Many people have ethical or religious concerns about the use of embryos and their loss of potential human life that could have been created from those embryos. Table 7. Table 8 shows information about some food components in cow's milk. So we can see energy in kilojoules per 500 cm cube, then fat, salt, calcium, and vitamin B12 in microgram given. And the recommended daily allowance of a typical person is also seen on the chart. How much more milk would a typical adult have to drink to get their RDA for calcium compared with the amount of milk needed to get their RDA for vitamin B12? So first of all, we need to calculate it for calcium, and then we need to calculate it for vitamin B12. So for calcium, we will divide the 500 with 605, and then we will multiply it with 10,000. So once we divide the 500 with 605, we will get the calcium per cm cube. All right, we will get the calcium per cm cube. And then after multiplying with 10,000, we will get how much calcium, we will get in how much milk we will need to get 1,000 milligrams of calcium. So the answer comes 826.45 cm cube. The answers are corrected to many decimal places because this is not the final answer. Then for vitamin B12. So B12. For B12, we can see in 500 cm cube, we have 4.5, but we require only 2.4. So once we do that, then we find out that our volume is needed 266.67. Now, to find out the comparison between the B12 and the calcium's volume, we'll have to do 826.45 minus 266.67, which will give us roughly 560. Answer corrected to two significant figure. If we correct it to three significant figure, it will be 559. So whichever is your choice, you write that one as the answer. Describe how a student would test cow's milk to show whether it contains protein and different types of carbohydrate. Okay, so for the test for the protein, first of all, the student should use a biorate reagent. The student will add the biorate reagent to the milk solution, and the solution will go from blue to lilac if there is protein present. Now, to test for starch present in the cow's milk, the student can use iodine solution, and by adding the iodine solution to the milk, the solution will go from orange brown to black if there is starch present. Now, if the student wants to test for reducing sugar, the student will use Benedict's reagent. So the student will add the Benedict's reagent solution to the milk, and then the student will heat it to 95 degrees Celsius, all right, or boil it, and the solution will go from blue to brick red if there is reducing sugar present. A scientist investigated the effect of bile on the breakdown of fat in a sample of milk. The scientist used an indicator that is colorless in solution with a pH lower than 10 and pink in solution with a pH above 10. This is the method the scientist used. Add 1 drop of the bile to the test tube and 1 drop of water to a second test tube. Add the following to each test tube: 5 cm cube of milk followed by 7 cm cube sodium carbonate solution to make the pH above 10, and then 5 drops of the indicator and 1 cm cube of lipase. Time: how long it takes for the indicator in the solution to become colorless. Now, solution with bile. Time taken for the indicator to become colorless in seconds: 65. Without the bile, it took a long time, which is 143. So we can definitely say bile increases the rate of digestion of lipids. Explain why the indicator in both tubes became colorless. Because the indicator was showing the color due to the presence of pH 10, which was made by sodium carbonate solution. Now, once the acid is produced, lipids are broken down, fatty acids are produced, which are acids. The acid then, basically, once the lipid is broken down by the action of the lipase enzyme, fatty acid and glycerol is produced. Fatty acid lowers the pH of the solution, and when fatty acid causes the pHs to become below 10, the indicator becomes colorless. Give the reason why the measurement of the time taken for the indicator to become colorless might be inaccurate. One of the main reasons why an observation of a color change can be inaccurate, it's mainly because the observation of the color change is subjective and more like it is based on opinion. So, because of that, we have to, since it is, it's subjective from one person to another. One person might say the color has changed, while the other person might say the color didn't change. That's why it can have a huge error. Explain the difference in the results for the two test tubes in table 9. So in the table 9, we know that one reaction was with bile. What is the job of bile? Bile emulsifies fat, meaning it creates larger surface area of the fat where the lipase can break down the fat to produce fatty acids more quickly. Guys, thank you for watching the video and best of luck for your exam. And guys, question paper videos like this will be uploaded every day. At least we're trying to. All right. So if you are watching the video and it helps you, then let the channel grow by suggesting it to your friends. All right. And if you require online tuition or something, you can let me know in the comments. My details are provided in the channel page. You can email me as well. All right. And that will help the channel to make more videos as well. So guys, thank you for watching the video. See you in the next video, guys. Bye bye.